Okay, we continue with our discussion of physical ways to control microbes. In this slide, you see a focus on what's called filtration. Filtration is exactly what it sounds like. If you had something that was contaminated with microbes, for example, a solution that you really need, maybe a pharmaceutical solution that you really need, but it got somehow contaminated with microbes, one thing you could do is pour it through a filter. So a filter is it's either a piece of paper or typically more like we call it a membrane, like it's made out of almost a little bit of a plasticky material, and it would have little holes in it, and you would choose the correct filter which had pores that would trap or prevent the microbes from being able to pass through, but the liquid pharmaceutical could pass through. And so you take your non-sterile medium, the contaminated mixture, you pour it through the filter, and only the pure medium would come through. And then you could probably put the filter in the biohazard bag. So this is just a simple way of removing microbes from a contaminated liquid. If air is contaminated, you typically use an air filter. So it works along the same premise. Air is moved through fans and things through rooms. And in our lab, for example, we have an air system that pushes all the air through filters. But in the hood especially, there is an air filtration system in the hood that moves the air through filters. And so this is always just to trap microorganisms in the filter itself. The filter can be made of different types of materials, but conceptually it has teeny tiny pores that will trap microorganisms, and the air can pass all the way through. One way to control microbes, especially in foods, is to use very high levels of salt or sugar or other things like that to make the water in the food become very hypertonic compared to the cells. And so the cells here, we're talking about the microbial cells. So microbes, when they find themselves in a hypertonic environment, will lose water and become dehydrated. And by removing that water, as I mentioned earlier, water is necessary as the solvent for the metabolic pathways that happen in the cell. So without the water, the cell will not be able to grow and probably will die over time. Fungus, though, actually do a little bit better in these cases. So especially with foods that have a lot of sugar, fungus actually does pretty well in those situations. But in terms of other kinds of microbes like bacteria, those would be inhibited by these solutes. So that's why people can cure their meat. We call it curing meat, and that's a way of preserving it to protect it from spoilage from microbes. And that's why we pickle things in a salt and a vinegar solution. And actually, the salt acts to increase the osmotic pressure, and the vinegar acts to change the pH, and the combination of those things and allows the vegetables to be stored without getting spoiled. And so it's a way of handling food contamination or reducing food contamination. All right, radiation. There's two types of radiation. One's called ionizing and one's called non-ionizing. Ionizing radiation has very small wavelengths. You may remember that energy travels in waves. Ionizing radiation, the wavelength is from the top of one peak to the top of the next peak of the wavelength, of the energy wave. And so when the wavelengths are very small, they're much more penetrating and much more damaging to cells. So if you really want to kill stuff, you use these ionizing radiation types. The problem is they're also dangerous to us too. But if you shoot electron beams, gamma rays, and X-rays at, for example, fresh fruit, what you'll find is that it kills the microbes and it doesn't stick around. So when you eat that strawberry, it's not like the radiation is still inside of it. It's gone by that point. If you don't blast it with the radiation, then the fungus and the microbes will cause spoilage. So a lot of the fresh foods that you eat are blasted with some kinds of radiation, usually X-rays, to make them, to kind of keep them from spoiling so quickly. Non-ionizing radiation are somewhat less penetrating. They can typically only hit surfaces. They won't pass through. They won't penetrate into the object. And so UV is a good example of this. It does not penetrate. So UV is good for disinfecting air, something that is clear or transparent, or things that are surfaces, but it cannot penetrate through them. So if you need something that's really penetrating, you would choose maybe X-rays or gamma rays. If something that's not penetrating is sufficient, then you could choose UV light as a source of radiation. So this chart here lists all the things that we just talked about. We talked about boiling, autoclaving, pasteurization, ultra-high temperature sterilization. You don't need to know the particular conditions, and we reviewed that all of these things have similar actions. So denatures proteins and destroys membranes, for example. And then you just need to know that boiling, autoclaving, pasteurization, and ultra-high sterilization are all types of moist heat, and what that means. And then for dry heat, you don't need to know the particulars, but just using like an oven, hot air, or a flame, incineration being total burning something up. And again, same kind of thing here. So we just learned the actions in general. You just need to know what the 4 actions are in general. And then I'm not going to ask you to match the particular actions to each of these types of methods. Refrigeration includes freezing. Desiccation, you need to know the definition of that. Lyophilization, so that has to do with cold temperatures. Filtration, air filtration and liquid filtration, what that means. Osmotic pressure, using salts and sugars to dehydrate things. And then the 2 different types of radiation. So you don't have to know all of this. And on this category, you just need to know the 4 in general that we listed in the first part of our lecture. But you do need to have a sense of these different methods in a kind of a general way, and the definition of what the words mean if they're not self-explanatory. Okay, now we move to chemical methods. In a similar way, we're going to have this list of things, and they all do the same actions that we talked about. They all disrupt cell membranes, cell walls, denature proteins, and disturb and destroy nucleic acids. The first one is called phenol, and chemicals that have phenol in them are phenolics. Phenol is a chemical. It's actually kind of nasty to work with. It doesn't smell very good, but it is historically used because it's a very good microbial control agent in healthcare settings, in labs, and so on. You will still see some of this in healthcare situations. However, as we have developed other kinds of chemicals that work pretty much as well and don't stink and aren't so kind of hard to handle, because these are pretty hard on human skin and such, we tend to go towards some other things nowadays. But you will still see some of these phenol-based products used as antimicrobials in a clinic, for example. Alcohol is probably one that you are familiar with. It is something that we can use. Our hand sanitizers have a fair amount of alcohol. You can swab skin with alcohol on a cotton ball prior to getting an injection, like a shot. So alcohol is good on surfaces, and it can be used on skin. Halogens, the term halogen, that's a term in chemistry that refers to iodine, chlorine, and bromine. And these are ions, but they are very good antimicrobials. So iodine, chlorine, and bromine can be used in different ways. Like chlorine, you put into a swimming pool to keep it from growing a bunch of nasty microbes. Iodine can be you can scrub your hands with iodine before. That's what doctors usually do before they go into surgery. Often there's iodine in those hand scrubs. Bromine, I have a hot tub and we use bromine tablets rather than chlorine tablets, just because that's what the manufacturer instructions told us to do. But they work the same. So they're very good antimicrobials. And when combined with oxidizing agents, you can get something like chlorine bleach. Chlorine bleach is actually very effective, should not be used on the human body, but is very effective in sterilizing and disinfecting surfaces and objects. All right. So, objects. So you can use iodine in your hand scrub. I would not scrub your hands with chlorine bleach, but it's kind of a combination of a couple different chemicals. But the chlorine part of it is of itself very antimicrobial, and then the bleach part of it is also, it's called hypochlorite. So anyway, these in red are called the halogens. All right. Oxidizing agents, you've probably heard of peroxide. So peroxide is also a type of bleach, but it's not a chlorine bleach. And you may have heard of ozone. So I put those here. There's other kinds of oxidizing agents, but these are these are antimicrobials, and they are disinfectants, and they can sterilize surfaces. Hydrogen peroxide can be used to sterilize an object. If you have a cut, though, if you're worried that it's infected, putting hydrogen peroxide on it probably won't do any good, because damaged cells will release an enzyme called catalase, which will inactivate the peroxide. But you can use hydrogen peroxide on skin that's not injured, and it will, well, I guess we'd say it'd be an antiseptic in that case. Ozone is actually a version of oxygen that's effective for treating drinking water, but it's expensive. So most cities will tend to use chlorine. They'll put chlorine in the drinking water at low levels. Of course, concentration is important, but at low levels, they put chlorine in the water. But ozone actually would be more effective. It's just more expensive. Surfactant